You have the different conditions, which are the substrate you have in tube A. The substrate is the food, as we said, the cooked starch. However, in tube B, you have the albumin. The digestive juice, you have the fresh saliva. This is the juice that contains the enzyme salivary amylase. Both of them are put on temperature 37 degrees for 1 hour. You see here it's 7, here it's 8, so it's for 1 hour. So the results after doing Fehling test for the first tube A, it gives a brick red precipitate, meaning that the starch has been transformed into the reducing sugar. So the brick red precipitates indicates that digestion takes place. So saliva acts on carbohydrates, which is something you already know. However, what we want to know, does saliva change albumin into, which is a protein, into peptides? So we have to do the Bio-Red test. So we did the Bio-Red test, it gives violet color. But as you know, in the table that I gave you, also peptides give violet color with Bio-Red test. So we have to add another test, which is the coagulation test, in order to see whether we have coagulation or not. If we have coagulation, then it's a protein. If we don't have coagulation, then it is a peptide. So if you perform a coagulation test on this, you will see that you have coagulation, meaning that you still have proteins. This indicates that fresh saliva does not transform proteins into peptides. So this means that in each enzyme is specific for a certain substrate. Each enzyme works on a substrate, on a definite substrate. It doesn't work on all substrates. Carbohydrates or starch is only transformed by saliva, by amylase. However, the proteins have other enzymes that transform them and work on them. I will ask you please to solve page 158.